Okay, I'm not going to talk about genetics too much, if that's okay. I'm going to talk about disruptive innovation, which Ian mentioned. But I'm going to talk about DNA sequencing. I'm going to talk about real-time DNA information and how the platform we've been developing, Oxford Nanopore, for just 12 years now, 12 and a half years, will actually allow us to take the amazing work that Anna's done and Michael and others are doing in translational research and cross the chasm into the applied markets. Let's see if I can make this thing work. So, to Ian's point, we are a DNA information company. As Ian said, disruptive innovation leads you to markets you do not think about at all. And right now, in industry, Mars are having a huge problem with salmonella infection in their factories. We've been approached by oil companies, bugs eat their pipelines. It's just amazing. Paint companies, infectious diseases are a problem in all these. Today I'm going to focus on health, as this is a precision medicine seminar. And what I want to talk about is the platform that we have and how we really think it can result in broad mass adoption, because that's what disruptive innovation should do. And if we look, for those of you who know this, apologies, but right now there are something like 8,000 to maybe 12,500, depending on how you count, DNA sequences in the world. So think mainframe computing. That's where DNA sequencing sits today. You're going to be spending $250,000, $1 million, several $1 million to buy these instruments. You need large infrastructures to run them and manage them. And they are amazing, amazing machines, but there are only 12,000. And you will not get broad adoption of some of the amazing research that in translational research into applied markets if you don't do something about that. That's where we come in. So I'm going to go quite quickly on what is nanopore sequencing. There's an amazing amount of resource on our website. But as I've said, DNA sequencing is like mainframe computing, and we all know what happened once we put power into our hands to be able to Google our health information. People go to the GP armed with a set of checklist systems. And at the beginning of that journey, everybody was very frightened that people would start to, particularly those who have a paranoia about their health, would just clog up all the surgeries. And actually, they turn up with checklists, they're better prepared, information really helps. We, and I will talk about the technology in a minute, we have a way of doing that in real time on a small device that can go all the way right through to something you can plug into your mobile phone, something you can do off a laptop, more complex machines for some of the whole genome stuff that Genomics PLC are doing to understand and bring these big data correlations together. So we are about to leave the mainframe moment, and we're going to do it with nanopore sequencing. Nanopore sequencing is pretty much, from a capital cost perspective, free. The devices cost less than $1,000. The commercial strategy for the business-minded people here is based on razor blade. There's a consumable in the device that sequences for a certain amount of time, and then you move on. Once you start to give real-time DNA information, there are a series of applications that are already enabled with this technology, and I'll give you a couple of examples. The information you get is phenomenally rich. It is incredibly long read length, and Anna mentioned structural variation in cancer. Understanding structural variation in long reads is critical. It allows you to look at modifications to DNA, so epigenetics or other base modifications, so it's incredibly rich in information. It's real time, it's really cheap, and our objective is to make it useful for anybody to do it. Right now, our library preparation, so that's taking some clean DNA, getting it ready for sequencing, takes 5 minutes, 2 steps. Any other sequencer requires a molecular biologist at grad or PhD level to manipulate that DNA over many hours or many days to get the DNA ready. Once you make it that simple, that low cost, you can open up to many, many more markets. So in one slide, and the prof would kill me, 25 years of work from Oxford, from Professor Hagan Bailey, on top of which 12 years at Oxford Nanopore. The technology, in its simplest embodiment, is a nanopore, a very small hole, existing nature. They're genetically engineered so that as we slide a double-stranded DNA, which is singulated with this green helicase, we get a processive, stepwise movement of the DNA. The charge, the shape and the size produce a small modulation in current flow. And that current flow, shown in this animation here, allows us to use complex signal processing to decouple this electronic signature into DNA bases. Now, just to give you an idea for the non-technologists in the room, we're talking about picoamps. So that's 1/10,000th the current going through a light bulb, 10 to 20 electrons we're counting. The size of the hole is 1/10,000th the size of a human hair. And we can pull all this together and we can put it in an array-based system. Because we are not using traditional methods which require chemical labels, sample prep is very simple. It means it can be taken out of a central laboratory. And because we're just sliding the strand through, as each strand is progressing, in real time, if you think about the last time you downloaded a film, how much quicker it's getting, and high-def films download really fast these days, we use the FPGA technologies that exist for downloading data. We can, in real time, stream that, we can deconvolute, and we can look at sequence pretty much in real time as it stands today. It fits into, I have a bag of tricks with me, fits into the MinION. I will pass it round. I would like to think most of you have seen it. It's got my name on it, so don't steal it. It basically has a small fan and a USB port that plugs into a laptop that provides software and power, and you can run that pretty much anywhere remotely. The flow cell is one of these. It'll do about 20 billion DNA bases before it runs out of steam, and then you replace that and you move on. So this device is the equivalent of having the computational power that you have a mainframe, which we have in our phones today. It really leaps that far forward. And commercially, it's been out there since 2014. We did early access for a year between 14 and 15. We've been selling it since then. It's in 42 countries. We have a couple of thousand accounts. People are using it for a whole broad range of things. In real time, you have to have something corrupt in the presentation, otherwise it's no fun. In real time, it will allow you to build these taxonomy trees. Is it beef? Is it horse? Is it drug resistant? Is there salmonella? What Mars want to know is there salmonella there in less than 2 or 3 hours? At the moment, traditional PCR diagnostics are an overnight test. They have to shut the factory down. It's costing them millions. And once you've got an infection, you never lose it. It's all about containment. It is never about completely eradicating. Although, if you were looking in real time, and you could trace it all the way back to the cocoa manufacturers, maybe you can. And that's the power of sequencing over conventional diagnostics. So, in the last 20 years, there's been a lot of talk about personalized medicine and precision medicine, and everybody thought at the dawn of the first whole human genome it was all going to unfold rather quickly, and there'd be these amazing associations between all the known genes, and voila. Hasn't quite played out like that, because the complexity and the associations between our whole genomes and diseases are so much harder to pull together. In addition, there hasn't been, although this chart would appear to show that anybody can do sequencing today, just to emphasize that point, you can do a $1,000 genome, but you have to be running that machine at full whack, spending multiple $1 million on consumables to get to those economies of scale, because these are batch-based, big, powerful machines. Again, they are not suited to precision medicine. They are not suited to personalised diagnostics. Now, you know I'm going to say we can solve all of that, and I am going to tell you how. Just a couple of quick examples. So, this product has been validated by NASA, and it's not so much that NASA is going to be the biggest customer. They're pretty rigorous, and they've taken it up onto space station, and it worked on there. So if you want to talk about field portability, it doesn't get much better than that. It's been used in the Antarctic. It's been used in various other settings. This is the amazing work Nick Lohman did, where they were looking at, this is a World Health Organization retrospective map of how Ebola spread. With this system, we can now start to do that in real time. And if you can pick that infectious disease out in real time, and you can see how it's mutating, how it's changing, you can start to contain and control. It will have a profound impact on infectious disease management. And in fact, Nick's gone on to take that same protocol and process flow, and they've done some amazing work on tracking Zika, which is on the front cover of Nature last week, I think. So with this technology, 10 minutes is not very much time. With this technology, you can map infectious diseases. I've already said this, you can use really, really long reads to look at structural variation in cancer. You can do UTIs, big problem. Takes too long. In the USA, the I'm going to have a go now as well, I'm mispronouncing something. Chikungunya virus and Hep C are real problems. And being able to do them rapidly in real time while the patients are still there and keeping hold of them is really important. Great example of how next-gen sequencing can really start to help and really usher in the personalised precision medicine age. This is diagnosis of a brain tumour on the same day. It only came out 2 days ago. Over conventional microarrays, you can look at methylations and modifications, you can structural variations, because you're doing something, I'm running out of time, you're doing something that you cannot do with other platforms. And that is, this is as close as it gets to McCoy's scanner. It can be hypothesis free. You can do a metagenomic scan of your DNA versus, I think it's this and this is what I'm looking for. So it gives you that richness. Now, the challenge is, once you've done this amazing work and you've got some panels and associations, how do you cross the chasm? And I'm going to spend the last couple of minutes talking about that. So once we have a known set of associations that link us to a disease and we want to be able to diagnose it, I do not believe current NGS, mainframe computing, will ever cross the chasm into the applied markets. In order to do that, you need a specific, application-specific NGS box, if you like. And this is our strategy for MinION-DX. So once this translational research garners these amazing panels, associations, areas of genes we should look at, to make them into diagnostics, we need single-use panel tests, multi-panel tests, single gene tests, whatever the genetics are. And because we can do it in real time, we can use software to pick out, if we pick HIV as an example, right now there's a PCR diagnostic that allows you to do a 2 kb region, which allows you to work out which drug resistance you have and which cocktail you need to be given. We can sequence a whole genome. If you can do it for the same price, you would. But this sequencer will not sequence other things. because a clinician only wants to have an answer to the biological question he's asked. Put this on an NGS box, 3 days later you've got a whole plethora of information, and the FDA and regulators don't know how to cross the chasm. So we strongly believe that moving from translational research to sequencing diagnostics will come from a very targeted approach, which is conventional, almost similar to how conventional diagnostics work today. Beyond that, you will all be measuring your information in exactly the same way as you Google your health, your symptoms. You will do the same. You will trend and track in real time because you can. And you'll turn up at the GP with a data set. Now, you may never see that data set. I have, this is a smidge iron. This is in development, plugged into a phone. You'll be able to do a very simple bacterial or viral screen. So you will all screen at home. We're going to have to do this. We cannot continue to just go to the GP and get antibiotics. It's going to kill us all. That alone, if you started screening at home and it was easy enough, it would save the NHS $2 billion a year. And it would probably allow us to start doing something about antibiotic stewardship. I'm out of time. That is my last slide. So if you take anything away from this talk, there's about to be a huge platform shift, and it will help to catalyse the revolution into personalised and precision medicine. Thank you for your time.